Hello students, heartily welcome on PARAVET digital platform. Today we are going to study cytology, a starting chapter of anatomy and physiology. Cytology, or we can say cell biology. What is cytology? Cytology is the study of cell. Cytology is the study of cell. And the other term which is commonly used, cell biology. Cell biology is a broad term which includes biochemistry, genetics, physiology of the cell, including study of cell, means study of cell in broad way. Its physiology, biochemistry, and also genetics. Now we are talking what is cell. Cell is the basic structural and functional unit of living beings or a body. Cell is the basic structural and functional unit. Functional unit of living beings. In other words, we can also say cell is the building block of body. In multicellular organisms, there are many of cells in number of trillions. Cell, according to cells, the animals or organisms divided into two types. One is unicellular and the another is a multicellular organism, like human beings. And in the veterinary, maximum animals are multicellular. Unicellular organisms are mainly, we study protozoology, and the lower category, lower structured animals like protozoa, means building block of multicellular organism. Cell is the building blocks of multicellular organisms. Now, the history of cytology. We are studying the history of cytology. At first, in the year 1665, a scientist naming Robert Hooke, who take a cork and make it slices and saw in his own microscope, and he observed that the cork is made up of tiny hollow spaces, means honeycomb like structures. Let's, in this image, Robert Hooke was the scientist who discovered his own microscope, and with the help of this microscope he observed wooden cork slice and saw this type of teeny hollow structures, and he gave a name this type of structures cella, and the cell name is derived from this word cella. The name cell is derived from this cella. Cella means hollow space. Robert Hooke does not discover living cell. He discovered only dead cell, because in the dry wood there are the cells having only cell wall, not cytoplasm, because cytoplasm after drying shrinks and cell become dead. So the we can say Robert Hooke is the discoverer of dead cell, or discovery of cell, Robert Hooke 1665. Another scientist, Antony von Leeuwenhoek, who discovered living cell. Means if the question arises, cell discovered, we will give answer Robert Hooke. If examiner asks us living cell discovery, then we will answer Antony von Leeuwenhoek. Now, discovery of cells. In 1665, scientist Robert Hooke used a microscope to examine thin slices of cork. He observed that cork is made up of tiny hollow compartments. The compartments reminded him small rooms, so he gave rooms the name cell. The plant means Robert Hooke discovered a plant cell or dead cell. Only cell wall was there. Means dead cell discovered by Robert Hooke. So now father of cytology. Father of cytology, which scientist? Robert Hooke. Father of cytology, Robert Hooke. And father of modern cytology, father of modern cytology, C.P. Swanson. Means Robert Hooke saw hollow compartments, and the name cell is also a Latin word which comes from chitos. Which comes from chitos. Chitos means hollow space. Latin term. Father of cytology, Robert Hooke. Father of modern cytology, C.P. Swanson. Cell is basic structural and functional unit of body. We have already studied that cells makes tissues, and tissues makes organs, and many organs makes organ system. Organ system makes the whole body. Some important points for examination, like to enlarge, to see in large way, the objects are seen with the help of microscope and instrument. Whenever the development of microscope Uh, According to this, in microbiology, there was progress according to development of microscope. Now we are talking about different type of microscopes. First is simple microscope, which is a glass, common glass, means convex lens, which enlarges the small organisms, small objects in a limited manner. Then compound microscope having two lenses, one is eyepiece and another is a objective lens. And the most advanced form of microscope is electron microscope, which was discovered by Knoll and Ruska. The compound microscope was discovered at Jensen and Jet Jensen. So the main important points occur on the basis. First is compound microscope discovery at Jensen and Jet Jensen. Second question is electron microscope discovery. By Knoll and Ruska. Electron microscope having a resolving power of 5 angstrom. Electron microscope is most powerful microscope which can makes the small object up to 2 lakh to 5 lakh times bigger. Electron microscope resolving power is in the range of 5 angstrom, which is very small. 1 angstrom is equal to 10 raised to power minus 10 meters. 1 angstrom is equal to 10 raised to power minus 10 meters. We are seeing the microscopes in this diagram. It is uniocular compound microscope. It is binocular. Both the eyes binocular microscope, and this is electron microscope. Electron microscope. Compound microscope having two lenses. This is eyepiece and this is objective lens. The magnifying power of compound microscope is the capacity by multiplying both the lenses, means magnifying power is multiplication of both the lenses. This type of microscope discovered by H. Jensen and J. Jensen. This microscope discovered by Knoll and Ruska. Now we are talking about largest and smallest cell. Largest cell, which is largest cell in animal kingdom? Ostrich egg. Largest cell of animal kingdom. and smallest cell is PPLO. PPLO means the full form is pleuropneumonia like organism. It is a type of mycoplasma. A mycoplasma. As we know, mycoplasma is a prokaryote organism having no cell wall. Now, longest cell is nerve cell of, also we call neuron of neck of giraffe. Giraffe, the neuron of neck of giraffe, the longest cell of the animal kingdom. Now, the living cell discovered by Antony von Leeuwenhoek. So, we are studying about living cell. What is living cell? Living cell in which cell having all the activities, physiological, biochemistry, and all the activities are properly maintained, which is called living cell, like spermatozoa, like bacteria, like protozoa. All of these cells discovered by Antony von Leeuwenhoek. Antony von Leeuwenhoek. This scientist, we are talking about Antony von Leeuwenhoek in year 1674. He used his own microscope. This microscope, Antony von Leeuwenhoek scientist used this microscope. It was simple microscope, and he viewed the pond water, teeth scrapings, and also spermatozoa and living organisms. These are the things Antony von Leeuwenhoek observed, like blood cells, white blood cells, red blood cells, yeast cells, bacteria, sperm cells. So, the main credit of discovery of living cells is given to Antony von Leeuwenhoek. He discovered bacteria, he discovered protozoa, he discovered spermatozoa. So, the scientist is known as father of protozoology, father of bacteriology, and also father of microbiology. Now we are talking about structure of cell. In veterinary, because we studies the animals, domestic animals, so we are studying animal cell. In this diagram there is animal cell. The outermost covering of animal cell is made up of cell membrane. Cell membrane, because of flexibility of cell membrane, animals having the body flexible rather than plants. Plants having cell wall as their outer covering on their cells. Both of these are animal cells. This cell surrounded by cell membrane. The outermost covering of animal cell is known as cell membrane. In the center part or middle part of the cell, there is a large cell organelle we know we can call it nucleus. Nucleus is the largest organelle of cell and main the controller, the manager of cell. Nucleus is bilayered and between nucleus and cell membrane the fluid portion which is called cytoplasm. If we include nucleus in this cytoplasm, we can say this all the structure, this whole the compartment protoplasm. The other organelles like mitochondria, endoplasmic reticulum, lysosome, Golgi body, and many other cell organelles in the cell presents in living cell. Now, according to one by one, step by step, we are going to study the cell organelles. We are starting from cell membrane. Cell membrane. Cell membrane, as I told, cell membrane is the outermost covering of cell. Cell membrane discovered by Nageli. Discovery or discovered by Nageli. Nageli and Kramer, there were two scientists, but Nageli is most popular. Cell membrane, the outermost covering of cell, animal cell. The outer most covering of animal cell known as cell membrane. Cell membrane protects the cell organelles, means the covering which protects various cell organelles which are soft, and the thickness of cell membrane is about 75 angstrom. Cell membrane, the structure was given by many scientists and many models in of cell membrane given by various scientists, like unilayer model. Unilayer model, a scientist Robertson give this model. Cell membrane's second model, important model, is trilayer, or we can say sandwich model. Sandwich model was given by Danieli and Dawson. And the most widely accepted model, widely distributed, widely popular model was fluid mosaic model, which was given by Singer and Nicholson in 1972. So according to various models, cell membrane classified cell membrane gives its advancements. We will study in next class with the structure, because of cell membrane is made up of phospholipid and protein. The bilayered phospholipid layer, which is in which protein molecules are embedded in two types of protein molecules, extrinsic and intrinsic protein molecules. So in next class we will study cell membrane and the other cell organelles step by step. Thanks.